Hello, my name is Basti Thiel, and today we're going to talk about metagenomics. So I'll take you all the way from profiling experiments, where you can profile the functions or organisms, species, taxa in microbial communities or microbiomes, all the way through to genome-based ecology, so how we can use genomes inferred from metagenomics to say something about the processes that are happening in the microbial ecosystem. So that's a lot of topics, and these topics are listed a little bit here. I will talk about metagenomics and its use in microbial ecology, the classical questions that we can ask by using metagenomic data. I'll talk about interaction between microbes and inferring interactions from co-occurrence of these microbes across samples. I'll talk a little bit about the interpretation of microbes in terms of their functionality and the evolutionary variability of these functions in microbial organisms. Then we'll discuss how to get genomes from metagenomic data sets, and then how to use these genomes to make inferences about the ecological functions of microbes. So like I said, a lot of topics. Microbiomes are ecosystems of interacting microbes, viruses, and microbes and viruses. And the human microbiome is perhaps the most well-known example. So there's a lot of important journals that have been publishing on the human microbiome. Basically, the message is that we are walking ecosystems that contain all these different microbes and viruses. More microbes in our body than human cells, and even more little viruses infecting all of those microbes, as well as sometimes our own human cells, of course. So this is all the organisms that comprise the human microbiome. But of course, the microbiome is not unique to humans. Every environment knows its microbiome. For example, the ocean has tons of microbes in it. But also the organisms within the ocean have specific microbiomes. We've looked at sponges before, and the corals, etc. And actually, there's a lot of microbial ecology that's going on in these microbiomes. But of course, there's a difference between the classical sort of ecology, where you can actually see the organisms that you're talking about, and microbial ecology, where these organisms are microscopic and thus invisible to the naked eye. Questions need to be answered in a different way. So exploratory ecological questions often talk about community diversity, such as the richness or evenness of a community. So we can see here in this forest at the top, there's 4 different species of trees, and they're evenly distributed because all the trees occur at equal frequency. In the second picture, we see that the richness of the ecosystem, of the forest ecosystem, is the same. There's still 4 species, but here there's a highly skewed distribution of species, and so the evenness is not very high in this case. These kinds of questions are sort of traditional questions that you can also ask about a microbial ecosystem, but to do that based on, for example, microscopy would be inaccurate, because a lot of microbes that might look the same under the microscope, as you can see here, might actually be different in terms of their genomics and of their functionality. So the best or easiest way to actually address questions like these ecological questions over here would be to sequence the DNA of this microbial community and try to infer which organisms are there. And then you can address these richness and evenness questions, ecological questions. So this extracting of DNA from these microbial communities is done with metagenomics. Metagenomics basically means sampling a site of interest. So the sponges over here, or the ocean water. Human gut is a very much sampled microbial environment. Then the microbes are filtered out. You can use different filter sizes to select, for example, bacteria or viruses or other size fractions of the DNA that you are interested in. And then once the microbes are filtered out, the DNA is extracted. Sometimes it's also amplified, although that does skew the abundances of the microbes in the original sample by amplification. Some things get amplified a little bit more, others get amplified a little bit less. So if you're really interested in the relative abundances of different organisms in the community, you should not use amplification because it affects the richness and evenness statistics. And then the DNA is sequenced on a nucleotide sequencing machine. This one is here a 454 Pyro sequencer. These don't exist anymore, or at least they're not supported anymore. There's now more modern machines. I'll have a picture on the next slide, I think. Yes, I do. And then, well, either way, once this random DNA sequencing has happened, you have therewith created a random sample of the DNA in the microbial community. Different parts of the DNA of these different organisms are amplified. And this is, because it's sort of a very large-scale random sample, this is thought or assumed to reflect the microbial community to some extent. And the analysis of these reads by bioinformatics is actually where the real fun starts, because then you can start to interpret what was actually going on in the original microbiome. So like I said, the modern machines are not the 454 Pyro sequencer, but there's very high throughput machines like this HiSeq X10, for example, where you can sequence 6 billion reads in a single run. And so that's basically, yeah, for $1,000 it says here you can sequence 30 times the human genome, or 30 times coverage of a human genome. And these prices are continually dropping. So what does this data really look like? This is actually the picture here at the top. This starry sky is what it looks like in the machine. There's a camera that actually captures this image and translates it to DNA sequence. But then once it comes into the bioinformatician's lab, this is actually what the data looks like. You get nucleotides at every position, a different letter of the nucleotide alphabet. And each of these letters is accompanied by a quality score that indicates how well this letter is actually been measured. So if it's perfectly measured, then it gets a high letter. If it's not that well measured, then it could get a lower letter, or sometimes different types of characters. And there's all kinds of bioinformatic programs that can interpret this kind of data and tell you what the most likely sequence is of every sequence that has been sequencing read that has been measured. So this kind of data is often deposited in public repositories, big databases that contain metagenomes from many different biomes: soil, marine, forests, forest soils, non-human hosts, human hosts, gut, grasslands, etc., etc. There's many different biomes that have been sampled, and EBI Metagenomics is one of the big databases. It's now called Magnify, actually, so this is an old logo. And the MGRAS database is another database that collects metagenomes, and also these databases allow preliminary analyses of the data, so you can answer already some of the basic questions. So what are these basic questions in metagenomics? Well, first question that is always asked is, who is actually there? Who has been measured? What are the organisms that these sequencing reads are derived from? So on this slide, I've got, this is one of my favorite slides. It's got a big bag of confetti on there, which actually illustrates the sort of the party that you get into your computer when you download a metagenomic data set from one of these databases. But it's not a real party yet, because all of the little pieces of confetti are still black and white. You don't know which organism they're from, and it's necessary to provide some color to this confetti before you can start to answer the question, who is there and what are they doing? These two questions are the classical questions that have been posed by Jo Handelsman already in 2004 in a seminal review that she wrote to discuss the promises of the metagenomics field. So who is there means that for each of these sequencing reads that you get, which are, like I mentioned before, a sample of the microbial community, you identify what organism it's from and what are they doing. You can answer that by by looking at the gene function that is encoded on one of these pieces of DNA. So the advantage with metagenomics is that it samples microbial organisms, and microbial organisms, including viruses, archaea, and bacteria, have very limited intergenic space. So most of the DNA that you sequence is actually coding for some kind of function. So that means that in a metagenomic data set, most of the data that you will be measuring is actually coding for a certain function, and you can really try to address the question, what are they doing, by looking at the functions encoded on each of these sequencing reads. How does that work? The simplest approach to do this is by using a big database. And so you might all be aware of the BLAST database at the NCBI in Maryland, where you can upload a sequence and you can identify which sequence in the big BLAST database, for example the nucleotide collection over here, that is the database that we're going to search against in this example, is most similar to the sequence that you put in. So if you would put in a sequence from a metagenome over here, then you could say, for example, this is my query sequence, it's coming from my sample. What is the sequence in the database that is most similar to it? And it could, for example, be a sequence from an E. coli bacterium, and it could encode the function RNA polymerase, because if you're lucky, the sequence that is most similar in the database actually has these annotations. It has an annotation of the taxonomic origin, so what organism is it coming from, and it could also have an annotation of the encoded function. Of course, with real metagenomics data sets, and especially the ones that are that consist of 6 billion reads, the BLAST algorithm that I showed on this slide is actually not sufficiently fast to deal with this kind of massive data. So with metagenomic analysis, you would use a different search tool, and these search tools are actually evolving all the time. So the latest one I can't tell you right now because it depends on when you're watching this video, but this is just an illustration. So this allows you, if you do this for all of these 6 billion reads, this allows you to actually get a profile of the taxonomic groups. So for example, at the level of phylum, you could indicate in each of the different samples, for example in the nose or in the ear or all of these human body sites, you could indicate which bacterial phyla are present and their abundance. So these are called stacked bar charts, where you have a bar chart that adds up to 100% of the data. 100% of the data here is listed on the y-axis, and the colors indicate which of the phyla are present. And you can also do a functional analysis where you measure which metabolic pathways, for example, are identified among the sequencing reads. And generally, 100% of the data here is 100% of the reads which could be classified. You should always remember that there is that not all the sequencing reads in a metagenomic dataset might be classified. So there's often also an unknown fraction that is actually still on top of this 100% bar chart. You can imagine that there's still more data that is not been classified by these search tools. And sometimes that can be a very large fraction, and sometimes there can be really interesting stuff happening in that fraction of unknown sequences. But we'll talk about that another time. So anyway, like I mentioned, you can identify which phyla and which functions are present in these metagenomic datasets. And if you want to sort of cluster the different metagenomes based on this composition of the microbiome, you can get really interesting pictures. You can do this, for example, by principal component analysis, where each of the different dots in one of these principal component plots represents a microbial community, and it represents in this case, the taxonomic profile of this microbial community. And you can see that all of the taxonomic profiles in the mouth, for example, look more alike than the mouth samples do to, for example, the gut samples, or to the urogenital tract samples, the skin and the nose. They overlap a little bit, so it looks like the skin and nose maybe harbor kind of similar microbiomes. Yeah, there's clear differences in the microbial communities in different human body sites. This is of course, an expected result, but it's really nice that it can be visualized like this. Another way of looking at these kind of samples, there's these stacked bar charts, there's these principal component plots. Another way that these are often displayed is by using a heat map. And in a heat map you can have all of the samples as rows, for example, or as columns, but in this case as rows. And the different taxonomic groups that you want to identify are listed as columns. And then the color gradient indicates the abundance of each of the different taxa in the different samples. And then the samples could be clustered. So for example, you can identify here a group of samples where these taxa are more abundant and these taxa are less abundant relatively, versus another group of samples over here, as you can see by this clustering dendrogram, where these taxa at the end of the set of columns are more abundant and these ones are less abundant. So you can see that there's two different kind of groups of samples in this data set, where some samples are enriched for these taxa and other samples are enriched for these taxa. So you could imagine that these might be, mouth-related samples and these might be gut-related samples, for example, because we saw on the previous slide already that the mouth and the gut harbor different microbial communities. So you can compare these samples by their taxonomic communities and cluster them like this. This is one way of looking at this data set. But of course you could also cluster the columns in this data set. You can also compare the taxa that are in the columns over here by their abundance profile across samples. And this allows you to identify groups of taxa that actually have a similar occurrence pattern. So these taxa over here, for example, they're very different than all of these taxa over here, because these taxa tend to occur in these samples and not in these samples. And these taxa over here occur more in these samples, where there's a difference actually between these guys over here that are in this top group of samples and the ones in the middle that are sort of distributed in all the samples a little bit. Yeah, so this kind of clustering approach, because it's just, vectors of data, you can cluster the rows, but you might as well cluster the columns too. And this allows you to really identify which taxa are actually co-occurring. And potentially, this could mean that these taxa are actually interacting in the ecosystem. And this is, of course, an important ecological question. How and which organisms interact actually, and how does this interaction work? So towards that, answering that question, clustering the taxa based on their abundance profile across samples allows you to make first guesses at which taxa might at least be interacting. This was really nicely done across a large set of metagenomic data sets already in 2010 by Samuel Chaffron, and he looked at all of these different taxonomic groups and identified clusters of taxonomic groups that tended to co-occur across different samples. And then you could, by looking at which samples they actually occurred in, he could label each of these clusters. For example, these ones occur together often in lake, water and pond samples. These occur in springs. These might occur in wastewater or bioreactors. And so each of these different clusters could be labeled with not only which taxa they were, because each of these little balls is of course a different taxonomic group, but also where this group of taxa co-occurs. And potentially this means that these are microbes that are actually interacting in these natural ecosystems. So I have a PhD student, or had a PhD student. He's now actually a postdoc in Spain, Felipe Coutinho, and he used this approach of clustering to analyze 180 marine metagenomes. He sampled most of these metagenomes actually from the MGRAS database that covered the different global ocean sites, as you can see with the yellow dots here in this map. But off the coast of Brazil, where Felipe comes from, there weren't any samples that were taken at that point. So we actually sampled 2 transects over here, as well as a couple of locations by the coast. And together collected 180 marine metagenomes, where we identified the bacterial genera that were present. So it's already quite interesting if you look at where each of these genera occurs. So these are all of the different bacterial genera that were detected. And you can see that there's a pretty good correlation in the ubiquity, so that's the number of samples where these bacteria occur, and the abundance of the bacteria, the average abundance of these bacterial genera per taxon. So there's a whole bunch of bacteria that are that might be considered generalists because they occur in many of the samples and also at a high abundance. Excuse me. But there's also several specialist genera. So these are genera that occur only in a few samples, but relatively abundantly. So it's kind of interesting. So then the next question that he asked is, why do these specific bacteria, each of these genera separately, why do they occur in the samples where they occur? Is there any environmental factors that are driving their abundance specifically in some of these samples? So in the samples off the coast of Brazil, Felipe actually measured a range of environmental parameters such as nutrient contents and temperature and the depth and the location on longitude and latitude, the coordinates. And each of these parameters were correlated with the abundance of each of the different bacterial genera to try and identify whether any of these parameters could be driving the abundance of each of these bacteria. And he found no significant correlation. So there were no significant correlations between any environmental parameters and any of the bacterial genera. Surprisingly, perhaps suggesting that none of the environmental parameters that were measured were of significant influence on the abundance of these bacteria. This was of course a little bit surprising, but we didn't give up, so the next thing that Felipe did was actually to try and correlate the abundance of each of the bacterial genera with the abundance of the other bacterial genera in the data set. And by doing that, he could identify clusters of bacteria. Each of the little dots in this, or the little diamonds in this plot indicates one of the bacterial genera. And he could identify clusters of bacteria that correlated really well with each other. So that means that all of these bacteria in this group, for example over here, actually correlates well with the abundance of the other bacteria in this group, suggesting that this is a sort of a community of bacteria that could be present together in the same kinds of samples. Green lines indicate positive correlations. Red lines indicate negative correlations. So the bacteria in this cluster of genera actually does not tend to co-occur with the bacteria in this cluster of genera. These bacteria are alpha proteobacteria and Bacteroidetes. This cluster contains many different types of bacteria. There's actually 11 different clusters that he could identify in the data set. Some of these correspond to taxonomic groups, like I mentioned here, alpha proteobacteria, Bacteroidetes, beta proteobacteria. So there's 2 clusters of cyanobacteria here and here. But some clusters also corresponded to expected functions of these different bacterial groups. Extremotolerant bacteria, ammonia-oxidizing archaea, for example, those groups. And then when he looked at the correlation between each of these different groups of genera with each other, he could identify really interesting patterns. For example, you already see here the negative correlation between this high diversity cluster over here and this alpha proteobacteria and Bacteroidetes cluster. Negative correlation. That's a negative correlation here, indicated with a red color between this cluster and this cluster. But there's significant correlations, positive as well as negative correlations, between many of the different clusters that we see in this full data set. So there's 11 clusters, like I mentioned, and a lot of them have positive or negative correlations with other clusters in the groups. This suggests that these bacteria within a cluster might actually form sort of a yeah, potentially interacting group of organisms. And if you are interested in these interactions between organisms, this is a very nice review that indicates the different types of positive, negative, or neutral interactions that there can occur between different types of between different organisms, specifically focused on microbial interactions. Next, what Philippe did was to take the abundance of each of these different bacterial genus clusters and then correlate the entire cluster with the environmental parameters. So here we see the environmental parameters again, and here we see the 11 clusters of genera, and you can see that now the colors indicate positive and negative correlations that are significant, and in contrast to the individual genus correlations, which did not yield any significant correlations, he did find a lot of significant correlations between the different clusters and the environmental parameters. For example, this high diversity cluster was correlated positively with depth, so it occurred at greater depth generally, whereas this cluster 5, which is the alpha proteobacteria bacteroidetes cluster, correlated negatively with depth, so those were probably more found at the surface of the ocean. So this shows that these groups of taxa are actually together indeed affect the abundance of these groups of taxa, is indeed together affected significantly by these environmental parameters. And this is at least a partial explanation for their correlation. So the correlation of the different genera in one of these clusters are together driven by the environment. So if these X and Y would be two different genera within a given cluster, and Z is an environmental parameter, then the effect of Z could influence the abundances of genera X and Y, and this could lead to the correlation between genera X and Y, and thus to infer the interaction between genus X and Y, even though they are correlating, could be due to an external parameter, such as the environmental parameter Z. This is a caveat that should always be kept in mind when interpreting correlation networks between bacterial organisms, such as the one that I showed 2 slides ago. But they do, and also 3 slides ago from the Chevron paper actually. Even though those correlation analyses do provide a preliminary information that might be suggesting an interaction between these microbes. So it gives you a hint, but you need additional evidence before you can accept the hypothesis of interaction. So as you can notice, I'm really careful with how I phrase this, and you should be too if you're interested in doing this kind of research. All right, because why? Well, we should. So one of the ways forward is to try and look at what the functions are of the different taxa that have been measured. So we measured a lot of different bacterial genera, like I mentioned, but then what is the function of each of these genera? How does each of these genera respond to the environmental parameters that we measured, or other environmental parameters, or even interactions with other microbes? This of course depends on the functions that are encoded on its genome. So there's a lot that can be said about the functions of different bacterial taxa. And this is because if you look at these functions, some functions are really stable in evolution. So for example, the light adaptation of different bacterial groups is a very ancient evolutionary trait. So a long time ago, this is an evolutionary tree here in black, a long time ago, the bacteria that are adapted to high light and to low light already grew apart, and so this is quite a stable taxonomic trait. This is for Prochlorococcus, for example. But more recently, different groups of Prochlorococci separated that were adapted to low or high iron, for example, or low or high temperature. And then the most variable trait in evolution is the adaptation to nutrient availability. So is there a lot of nutrients in the environment or not so many nutrients in the environment? And this is a trait that can vary really quickly and thus is not very easily predicted from the presence of these genera. So if you see this genus or this genus, they might look very much the same. You can't really predict whether the nutrients the nutrients of their environment are high or low. But if you see this genus or this genus, you can definitely predict the temperature, iron content, and light of the environment. This is a little bit about the variability of these ecological functions. And if you look at the overall functional profile of genome sequences, you can see that with increasing distance in similarity between genera, so this is based on the 16S rRNA gene, which is a marker gene for taxonomy. 100% means that these two organisms are basically the same species, or actually the same species is already accepted. The species threshold is already accepted at higher than 97% identity of the 16S rRNA gene. And as you can see, in all of this region, all of these little blue dots here are pairwise comparisons between the functional content of different genomes with between 100 and 97% identical 16S rNA genes. So all of these bacterial genome pairs are supposed to be the same species. What you can see is that some of these genomes have only 40% similarity in their functional content on their genome. So that means that the genomes are extremely different. In some of these cases over here, these might be outliers, but in general you can see that the similarity between the functional content of genomes of organisms that are supposed to be in the same species according to this threshold can be easily down to 70% or 30% difference, or 70% similarity in functional content. And this is exactly these kind of functions that are highly variable in evolution, right? So the adaptation to nutrition, but also to temperature and other environmental parameters might be very variable in these genomes. So if you want to guess the ecological function of a bacterial taxon, you really need to look at the content of the genomes before you can say what the function of these bacteria is, and before you can say why this specific bacterium actually correlates with another bacterium that you can also find in the metagenomic data. And then, yeah, to make things worse, we constantly discover new microbes and viruses, and so we constantly discover new compositions of the functional content of genomes that might affect the response of these microbes to their environment. So this is a quick picture of the microbial tree of life, or actually the entire tree of life. Over here, let's see, over here in the bottom right hand corner there's humans, other animals, other eukaryotes, plants, etc. And then this is the eukaryotic tree. These are archaea. Archaea are actually really close to eukaryotes. Eukaryotes just a sub-branch of the archaeoclade. And these are bacteria. And as you can see, bacteria are vastly diverse. And then these red dots indicate clades that have no cultured representatives. And everything that is not italic is actually not yet been characterized properly. So for all of these bacteria that are not yet characterized properly, including the ones where we don't have a cultured representative, we cannot really say what their function is because we haven't been able to measure it yet. In some cases, we can get the genomes out from the metagenomes, and we'll talk about that next. But first, quickly back to what Joe Handelsman said. So Joe Handelsman said, we only need to answer the questions, the traditional questions: who is there and what functions are they encoding? So this is still possible because we have these sequencing reads. We can identify for every sequencing read which type of organism it's most likely derived from because we just look at the sequence similarity. We can identify which function is encoded on this little piece of DNA. So we can make these functional profiles. We can say what taxa are there, who's there, and we can say what are they doing, what functions are encoded in each of these different environments. The problem is, of course, that we cannot say who is doing what. So to link the function to the taxonomy could still be a problem. If two functions are found within an environment, the question that we are still stuck with is, are they really connected? So we can find a metagenome with functions such as ears, antlers, wings, and teeth, but do we really, can we really make the assumption that all of these functions are actually connected in a single organism, or are we actually inferring a chimeric organism such as this Wolpertinker? That's such a nice picture over here. So to do that, we need to take the genes that we have identified within the metagenome. So for example, we identify gene 1 and gene 2. Gene 1 we know catalyzes the reaction from A to B. Gene 2 might catalyze the reaction from B to C. And we need to see if all of these functions are present in the same genome, or maybe they're present in different genomes. And in this case, we can infer that potentially there's an interaction between this yellow organism and this red organism, which might be a cross-feeding based on metabolite B that might go from this organism to this organism. So in that case, if we really can look at the functions of the genomes as individual genomes, we might be able to say something about the interaction between them. So to do that, we need to infer genomes from metagenomes, like I mentioned. So we need to go from this confetti kind of picture of the metagenomic data, of the microbiome, to a genome-resolved metagenomics analysis, where we try to assemble from each of the from the different blue reads, for example, we would identify the blue genome, etc., etc. Luckily, this is possible with advances in high-throughput DNA sequencing. So there's a vast evolution of second and third generation sequencing platforms, where we can get tons and tons of data with the HiSeq-X, like I already introduced above. But also the long-read sequencing platforms such as the Nanopore and the PacBio are being used more and more to analyze metagenomic data, because they allow us to really put all of these little short reads together into longer contexts that represent better fractions of the bacterial genomes. So we try to get from reads to genomes through de novo sequence assembly, of course. And if you're interested in how this works, I recommend watching another YouTube video that I have over here. So please click that link. I'm not sure, actually, the link probably doesn't work, but anyway, you have to type this code and you can see the lecture about assembly. But in the end, we obtain contigs, and then contigs, of course, are not yet genomes, so we still need to do binning. Binning is basically the clustering of contigs that are derived from the same genome into bins that represent bacterial genomes. So this would be contigs together of the red genome, the blue genome, and the yellow genome. And if you do this, then you can really start to look at which functions are identified together within this genome, which functions are identified within this genome and in this genome. And then you can start to try and make inferences about the interactions between these bacteria. To illustrate this, I will highlight the work of another former PhD student of mine. He's now at Caltech in California, Daan Spet, and he analyzed the microbial community in the wastewater treatment plant in Olburgen. There's a treatment plant that removes carbon over here, I think, and then phosphate over here, and then in the third step there's a special treatment box, which is an Anamox reactor, and that is there to remove the ammonia. How does it do that? It has a community that is dominated by two groups of important bacteria, and anaerobic ammonium oxidizers and aerobic ammonium oxidizers. The anaerobic ammonium oxidizers, the aerobic ammonium oxidizers over here in blue, take the ammonia and some of the oxygen that is present in the water and together convert this to nitrite. Then the anaerobic ammonium oxidizers over here in red take also some of the ammonia together with the nitrite that was generated by these aerobic ammonium oxidizers, and through a reaction called the Anamox reaction, converted to nitrogen gas, which is a clean waste gas and can be released into the atmosphere without any problem, and thus the ammonia is removed through a biological process involving two important interaction partners. So what we were interested in doing is to figure out which other bacteria are also there in this bioreactor. Of course, it was a large bioreactor, 600 cubic meters, there's much more present than just these two bacteria, and we were wondering how they influence this Anamox process. So to investigate this, Dan sampled the bioreactor, extracted and sequenced the DNA. In this case, he used an ion tolerant sequencer for that. Assembled the sequencing reads into contigs, binned the contigs into genomes, which were classified in the context of the rest of the bacteria that were known in the database, so the bacteria sequences from the database, and assessed the completeness of each of the different bacterial genome bins that we identified by using single copy marker genes. And this assembly, binning, and quality assessment steps are explained in more detail in the video that I provided the link for a few slides back. So the video about assembly and binning. So you can listen to more details about this technical stuff over there. And then he identified 23 draft genomes that we measured were at least 80% complete. So this was the cutoff that we used for saying that these genomes were complete enough to make strong enough statements about the functions that they encoded. And these 23 genomes are here shown in the context of the bacterial phylogeny. So the red ones are the ones that we inferred, and the black ones that are also in the tree are the closest other genome sequences that were in the database. And as you can see, in some cases, these genomes are quite similar to other known bacteria. For example, this Anamox bacterium is a planktomycete that is very similar to a Brocadia anamoxidans, which is already previously sequenced from an Anamox reactor. But there's also really new organisms that are actually deeply branching in the bacterial phylogeny. For example, this OP3 genome was the first of its kind. Well, I don't know exactly what level, but it was very different from bacteria that were already known. So we found a bunch of new bacteria. Many of these bacteria were interestingly previously also found in Anamox reactors, which is indicated by this black part of the pie chart here. That's the percentage of times that it was observed in other Anamox systems, and they represented more than half of the DNA in the metagenome, suggesting that more than half of the bacterial DNA in the microbiome was actually derived from these 23 genomes. That means that there's probably more rare genomes that are also present in the system, but those we did not take into account in our analysis. So we analyzed these 23 genomes and identified different genes that were responsible for different steps in the nitrogen cycle. So that included the aerobic ammonium oxidizing steps, but also the anaerobic ammonium oxidizing steps, and other steps of other important metabolic processes that were related to this nitrogen cycle. So here there's some, for example, some carbon metabolism steps as well. By identifying which of these genes were present in which of the genomes, we could classify the genomes into 11 groups. The first group being the Anamox bacterium. There was only one genome that was related to Brocadia sinica, which is actually the one that I showed you here at the top, is related to Brocadia anamoxidans. Sorry. And so there was only one of those organisms that could do that step. But there were multiple organisms, for example, that could catalyze some of the other steps in the pathway. For example, there's a nitrite reduction and detoxification group of bacterium that could be catalyzed by different chloroflexi. There's heterotrophic bacteria that could do denitrification. That's step number 5 over here. That takes some nitrate and converts it back into nitrite by using some organic carbon, for example. And thus these bacteria actually help to improve the nitrite pool once again, so that the nitrite and ammonia could be, through the Anamox process, converted into safe nitrogen gas. And this gave us a better view of how this Anamox reactor actually worked. Okay, so I hope you enjoyed this overview of metagenomics, all the way from sampling and profiling through to genome-based ecology. Yeah, thank you very much for listening.